So we've got lots of definitions in these slides. Anything in purple or bolded is probably a really good definition that you want to remember. So do keep them in mind. Make flashcards out of them, I recommend. So I use Anki in uni, but in high school I'd use like Quizlet, or I'd like make physical flashcards. So you can make like actual flashcards and print them out and use them, and that might be a really useful way of studying. So what is gene expression? A gene is a particular sequence of DNA bases which encodes for a specific polypeptide chain. So these are inherited from a parent to an offspring, and they're kept within particular loci or locuses. Loci is the actual plural form of that word. And so what a locus is a fixed position in the chromosome where a specific gene is located. What is gene expression itself? So this is the transcription and translation of a gene. And so this is the process by which genetic material such as DNA is converted into a functional 3D protein. So it goes from DNA to RNA or mRNA, and this mRNA will then be translated into a polypeptide, hopefully a functional protein. So there are many different stages of gene expressions. The first one is transcription, which occurs in the nucleus. So you have a pre-mRNA strand being formed from DNA. In stage 2, the pre-mRNA must undergo post-transcriptional modifications in the nucleus in order to become mature mRNA. The third step entails the mRNA moving to a ribosome. This can either be a free ribosome in the cytosol, or it can be attached to the rough ER. After this, mRNA will be translated at the ribosome into a polypeptide chain, and then finally that polypeptide chain will be modified, transported, and secreted or used within the cell. Cool, so this process here is just showing transcription. So you can see the DNA here is double-stranded. You've got the 5' and 3' end, you've got the coding strand and the template strand, and you've got the RNA polymerase, which kind of opens up. So it unwinds that DNA and opens it up to access it, and this allows the making of an mRNA strand. And you can see that's being made here. You can see that in DNA we've got 4 different nucleotide bases, A, G, C, and T, and in RNA we've got A, G, C, and U. And so these 4 bases are going to be used to create the messenger RNA. So I've kind of tried to summarize the content as much as I can. I know it's a pretty intensive topic and it's been quite difficult to get your head around it. So I'd recommend trying to work through this slowly in your own time as well. It also depends on your way of studying. I personally prefer to read textbooks or like thick slabs of paragraphs, whereas some people might prefer videos. So really figure out what works best for you. And if you are more of a video person, maybe take the time to look up on YouTube transcription simplified or something like that. So what is transcription? This is the process of producing mRNA from a DNA template. Okay, let me have some water first. Since the process occurs in the nucleus, what happens is that this enzyme called RNA polymerase binds to the promoter region of the gene to be transcribed on the template strand of DNA. You get lots of different enzymes in bio, and it's kind of hard to remember their names sometimes. But if you kind of break down the names, it kind of makes it a bit easier, I think. So RNA polymerase, well, enzymes typically end in A, so anything ending in A, you can kind of guess is going to be an enzyme. So polymerase enzyme. What is a polymer? So I find it really useful to look up the etymology of words in bio. So if you look up polymer, polymer means a long chain. And so if I've got polymerase, an enzyme which makes a long chain. And at the start, you've got RNA. So you can kind of presume that this enzyme is going to be something which makes a long chain of RNA, which is exactly what RNA polymerase does. It makes a long strand of RNA, mRNA. So what happens is that this enzyme will bind to this particular region of the gene known as the promoter region, and it will bind on the template strand of DNA. The RNA polymerase molecule will then unwind the DNA, and then it moves across the template strand and reads it in a 3' to 5' direction, while synthesizing RNA by joining ribonucleotides in the opposite direction, so the 5' to 3' direction. It says here, remember that strands are anti-parallel. What that means is that if you've got 5' to 3' this way, 5', 3', the opposite strand will have the opposite, so it'll be 5' to 3' that way. Okay, so it's kind of opposite directions, and that's what they mean by anti-parallel. So once that RNA polymerase reaches the end of the gene, or the termination sequence, the pre-mRNA molecule will be released. The pre-mRNA strand is complementary to the template strand and has the same sequence as the coding strand, except that T is replaced with U. This is kind of an intense looking diagram, but it really just does explain what we've just gone over. So we've got 3 stages here. You don't need to know their names in VCE, but do recognize what occurs. So we can see here the red part. In the nucleus, RNA polymerase recognizes the recognition sites, causing it to bind to the promoter, which is the side of the gene. The RNA polymerase then separates the DNA into single strands. The template strand can be read in the 3' to 5' direction. The pre-mRNA nucleotides are quickly paired with their complementary bases, which correspond with the template strand of DNA. The pre-mRNA moves in the 5' to 3' direction, while the template strand of DNA moves oppositely from the 3' to 5' direction. Honestly, I think this diagram just kind of is quite technical. It pretty much says exactly what we just discussed, but I think it's a lot more digestible in these little dot points. So I recommend going back and reading these ones. Okay, next up we've got RNA processing. And so eukaryotic cells will have 3 types of post-transcriptional modifications within the nucleus. Keep in mind that this is in eukaryotic cells, not prokaryotic cells. So in eukaryotes, we've got introns being removed and exons being spliced or joined together. What are introns? I think of them as interrupting regions of a gene. So they are non-coding regions, whereas exons are coding regions. And remember that exons are expressed. So exons express, introns interrupting. This means that mature mRNA is shorter than pre-mRNA because the introns will be sliced out. And I think I've got a diagram on the next slide where I'll show you this. You've also got a methylguanosine cap being added to the 5' end of the RNA molecule and a poly A tail being added to the 3' end of the RNA molecule. And once these modifications have taken place, the RNA molecule will become mature mRNA and it will leave the nucleus to move to a ribosome. So you can see here we've got that pre-mRNA will have the introns being spliced out and the exons, which are expressed, joined together with the addition of a 5' cap, methylguanosine cap, and a poly A tail, which pretty much just means a long chain of A's added to the end of that. And these kind of assist in export from the nucleus. Same diagram once again, just kind of showing it a different visual. So why do we remove introns? So it's really important that we remove introns because these regions do not code for protein molecules and they will be useless in translation. We do use these regions in some genome modification processes, but we don't really discuss this in VCE. So don't worry too much about it, unless you want to. So one gene may be able to code for more than one protein or variation of a protein depending on which exons are kept in or left out, and you'll have alternate exon splicing. So you can actually shuffle about these genes. You'll look at that if you do biomed in uni. I really enjoyed that and I thought it was super interesting. But yeah, we don't really look at that too much in VCE. This may also help explain why the amount of proteins in the body are so diverse despite the amount of DNA present. So we've got the same DNA in all of our cells, but different cells will express different proteins. So our skin cells will express particular proteins needed in the skin, whereas our liver cells will express different proteins. Our kidney cells will express different proteins. They all express different proteins. And that's because they kind of shuffle about the exons and have alternate splicing. So what is the point of that 5' methylguanosine cap? It's really important because it helps to initiate the process of translation, which is recognition at the ribosome. Both the 5' cap and the 3' poly A tail protect the RNA strand from damage from enzymes, and they also add stability. The 3' poly A tail also assists in the export from the nucleus. So that mRNA is going to leave the nucleus via a nuclear pore, and it's going to go off and travel to the ribosome. Protein synthesis. So this is the third part, translation. So translation is the synthesis of a protein, and it's the process by which a polypeptide molecule is produced from mRNA at a ribosome. So what happens? Once it leaves the nucleus, the mRNA strand will migrate to a ribosome. It will enter the ribosome at the 5' end, and the start codon AUG is going to instruct for translation to begin, directing for the amino acid methionine to start the polypeptide chain. Each successive codon in the mRNA will pair up with the anticodon of a tRNA molecule carrying a specific amino acid within the ribosome. The process continues with more codons and anticodons pairing, resulting in amino acids being carried by the tRNA molecules being added to the growing polypeptide chain via peptide bonding or condensation polymerization. And once a stop codon, so UAA, UAG, or UGA, which don't code for amino acids, are reached, translation will cease and the polypeptide chain will be released. I just realized this just says like 1, 1, 1. Sorry about that. These are all progressive steps. So keep that in mind. So here we've got gene expression occurring, we've got the ribosome reading the mRNA, and we've got our tRNAs bringing with them amino acids in order to facilitate that process of creating that polypeptide chain. Cool, hopefully that makes sense. Okay, in terms of gene structure, there are a few more words to know here. So I did mention before, introns are non-coding sections of a gene, or so-called interrupting regions. Exons are sections of a gene that form mRNA and code for proteins, or they are the expressed regions. And you've got the promoter region, which is a section of gene which RNA polymerase can bind to. Operator regions are found in prokaryotic operons, which we'll look at in a few slides, but these are not in eukaryotic genes. So how come different cells produce different proteins even though every cell contains the same genome? Well, pretty much cells can switch genes on and off, and this is called upregulating or downregulating. And so they can up or downregulate particular proteins based on the specific needs of a cell. So you can see here, in this muscle cell, we definitely need muscle protein, and so we might actually express gene A, which might be really important in the muscle cell. We don't need gene B perhaps, and so we can downregulate it, and we can upregulate gene C just a little bit. And this may be exactly what we need for the muscle cell, but you can see that the needs differentiate in the skin cell, and that we actually don't need so much expression of gene A in the skin cell. We do need quite a bit of B, so we'll upregulate B, downregulate A, and probably downregulate C as well. And then for the third one, you can see that you definitely downregulate gene B for the nerve cell. So really just kind of switching these genes on and off as needed in order to facilitate and kind of ensure that you're looking at the particular needs of a cell. So how are genes turned on and off? So we've got lots of different transcription factors, and what these are binding proteins, or DNA binding proteins, which bind to specific sequences of DNA at the promoter region and other regions as well, and they regulate gene expression, and they can either increase or decrease transcription of a gene. So things called repressors, these are transcription factors, and they prevent transcription by binding to the promoter region or the operator region of DNA, thereby preventing RNA polymerase from binding and transcribing the DNA. So you can see you've got activators binding to enhancer regions, and this leads to high levels of transcription. You may have repressors binding to operator regions, and this may suppress transcription and actually prevent transcription from occurring, thus downregulating a particular gene. So how are genes turned on or off? You've got a few other genes. You've got regulatory genes. These genes encode for the production of proteins, such as transcription factors, that regulate gene expression. And you've also got structural genes, and so these are genes that produce a protein or product that forms part of the structure or the functioning of an organism. Essentially every gene except regulatory genes. You've got signaling molecules. So signaling molecules such as hormones can bind to receptors on or within a cell, and they can influence gene expression in that particular cell. So a lot of signaling molecules such as hormones are actually proteins, and so they can activate transcription factors that assist in RNA polymerase binding. They can inactivate transcription factors that prevent RNA polymerase binding, can inhibit or switch off particular genes, and also bind DNA directly, and this can alter gene expression also. I'm like dehydrated. I need to get more water. So gene expression is influenced by cell type, stage of development, and the external and internal environment of a cell. Okay, so different cells can actually produce different protein products despite containing the same DNA, and so genes are only expressed when the product's needed. Why we do this is to save energy and resources. There's no point like expressing a particular gene and making proteins for something that is not required. Like if you're a skin cell and you're making liver proteins, you don't need liver proteins there. There is no point. So you want to save energy and resources by just turning that off and not needing to use it. We can produce excess proteins or proteins when they're not needed. When proteins are not needed by a cell and we're producing them, this can actually harm the organism. It can interfere with the normal functioning of the cell and kind of disturb it. Some genes are only required to be expressed at certain points in the lifespan, such as in embryonic development. So you've got these things called Hox genes, and they can help in digit formation. These are not really needed to be expressed later on in life, or it really depends on the needs of a person or individual or a cell type, but a lot of them are not required after you are born or after embryonic development is complete. So just turning them on would be completely wasteful and may also harm the organism. Some genes also need to be expressed in all cells at all times, and so, sorry, my voice is just jumping over my words. These are often referred to as housekeeping genes, and so some of them are actually really important and they will be constantly expressed, but some of them don't need to be expressed all the time. Okay, what's an operon? So an operon is a segment of DNA containing a group of genes that are transcribed together. So it doesn't exist in eukaryotes. Really keep this in mind, operons only exist in prokaryotes, okay? So it's a bunch of genes which are transcribed together. They're separate from the promoter where the RNA polymerase binds. So an operon consists of a series of structural genes which are transcribed together, only in prokaryotes. And so one method of gene expression regulation is a trip operon, and this is explicitly defined in your study design. There also used to be something called the lac operon, so I'd recommend looking that up as well. Lac operon and trip operon, but now your study design is going to be testing the trip operon, and so we'll look at that soon. So there is a gene in some bacteria, including E. coli, and this encodes for the production of the particular protein tryptophan, also known as TRP. When too much tryptophan is present, we have tryptophan binding to a repressor. What happens is that this tryptophan molecule binds to the repressor and activates it, and once it's in this activated conformation, it can then bind to the operator region of a gene, and this will prevent transcription from occurring and prevents RNA polymerase from binding to the promoter region. So when you've got too much tryptophan, it's kind of like a defense mechanism kind of thing. It's going to kick in and be like, look, too much tryptophan, let's activate the repressor. So it activates the repressor binds, and it prevents transcription. When there is little or no tryptophan, this means that tryptophan doesn't bind to the repressor, and it causes the repressor to actually change in conformation. The repressor will then detach from the operator region, and this allows transcription to occur, as RNA polymerase can then bind to the promoter of the operon. The repressor is coded for by a regulatory gene, TRPR, and this sits upstream, so just like way out, separate from the operon. And so it's way upstream of the tryptophan producing gene on the operon. So the repressor is also a protein or an enzyme. It's encoded by a gene because it is a protein, and it's just like separate to the entire operon. Okay, diagram. So this line here, it took me a really long time to know this or learn this, but that means it's like way away. Like it's not right next to it, it's actually like quite a long way away. So here is your operon. Trip E, D, C, B, and A are our structural genes. And then you've got a few regulatory genes. And you've got the promoter region where the polymerase wants to bind, and the operator region where the repressor will bind. And here you've got TRPL, which is known as the leader sequence. And we'll look at that soon. TRPR is way upstream. It encodes for the TRP repressor. And so what happens is that will be transcribed into mRNA. That mRNA will be translated into an actual protein. And so what that protein is actually the TRP repressor. And so here it is in its inactivated state. When you've got a high amount of tryptophan, it will bind to the repressor and activate the repressor. After this, like when the repressor is activated, it's actually going to bind to the operator region. And so in binding to the operator region, it causes a physical blockade there. And this actually prevents the RNA polymerase from being able to bind to the promoter region. As a result of this, the RNA polymerase can't actually go through and transcribe these structural genes. So you have kind of the entire operon just like being shut off. Like these genes are going to be downregulated because the polymerase can't actually bind the promoter region and can't actually go through and transcribe these genes. Okay, is it still recording? Oh my gosh, I thought it stopped recording for a minute. We are fine. Okay, so that was the first method of kind of like regulating the trip operon. So you actually have the repressor being activated. When the repressor is activated, it will bind to the operator region and prevent the polymerase from going through and transcribing these genes. We have a few more methods of regulating trip operon. So one of them is known as attenuation. And so, like, although repressors prevent transcription from starting, attenuation actually prevents transcription from completing. And so a region called the leader codes for an attenuated sequence. And the attenuated sequence forms hairpin structures. So within the leader are two tryptophan codons. This means that in order to translate the leader, we need tryptophan. This is kind of a complex process, so I'll try and work through it slowly. I mentioned we've got the TRPL here, which is the leader sequence, and inside the leader we've actually got an attenuator. So this attenuator forms a hairpin loop. So I might just open up a browser here and explain what I mean by that. Hairpin Oh, shouty capitals. Hairpin loop. So RNA we know is single-stranded, unlike DNA, which is double-stranded. What happens is that RNA can actually form loops in on itself. So you can see here that while it's a single-stranded structure, it's forming complementary base pairing with its own strand, right? And so it's forming lots of loops. And so as a result of this, you have the formation of hairpin loops. So what happens is that the trip-L sequence, which is the leader sequence, it will actually be transcribed into mRNA. Consequently, this mRNA will then actually go through and it will form some hairpin loops. And there are two types of hairpin loops. Something else to note is that within the leader, there are two tryptophan codons, and these codons encode for tryptophan protein. So in order to translate the leader, we need tryptophan. And so depending on the level of tryptophan we will have different hairpin loops forming. How are we going for time? We're fine. So in the case of low tryptophan levels, the ribosome, okay, I need to pause here. I didn't learn this until uni, this wasn't part of my study design. I had something called the lacquer one, which was much more simplistic. I found I had a lot of trouble learning this because it just took me, I don't think anyone really simplified this enough for me, but I didn't realize that in prokaryotes, transcription and translation can occur at the same time. So I show my students a diagram. So you can see here, you've got the DNA. In prokaryotes, there's no nucleus. As a result, transcription and translation can occur together because there's no segregation of these two processes. Because the nucleus means the ribosomes can't get in, and the mRNA is going to leave the nucleus first in order to be translated in eukaryotes, right? But in prokaryotes, there's no such thing as a nucleus. Sorry. So because in prokaryotes, the DNA and because the DNA isn't sequestered in the nucleus, you can actually have ribosomes going through and translating that mRNA straight away. So what you do is you have RNA polymerase reading the DNA and making mRNA. So this purple thing here is the mRNA strand. And pretty much as soon as it starts reading it, a ribosome is going to jump on and start translating that, okay? So pretty much transcription and translation are occurring simultaneously because there is no sequestering of the mRNA in the nucleus, because there is no nucleus. So it's really important to understand that transcription and translation occur at the same time in prokaryotes. Okay, so what happens is that you have that leader sequence being transcribed and it will form some kind of a hairpin loop. And if you've got enough tryptophan, you can translate the leader. What happens there is in low tryptophan levels, the ribosome will move through the leader really slowly as we need tryptophan for translation. As such, a hairpin loop is created that does not stop transcription, so transcription occurs. In high tryptophan levels, the ribosome will move through the leader very quickly as we have lots of tryptophan to translate the sequence. A hairpin structure is created that does not stop transcription, therefore transcription doesn't occur. So it does stop transcription, so therefore transcription doesn't occur. And as such, the ribosome folds up the mRNA and RNA polymerase detaches from the operon. This is all really complex. The way that I think of it is if you've got low amounts of tryptophan, when the ribosomes go through it, they've got to translate it slowly because they're looking for tryptophan. They've got to go through and search for that extra tryptophan, right? So it's kind of a slower process. So what happens is they go through and they read the mRNA slowly. And they read it very slowly. So they're quite calm and relaxed. The way I think of the other process though is in high tryptophan levels, you can quickly translate, you zoom through it, and it kind of creates a formation of a hairpin loop, which in my head I like to think of it as it zooms through super fast, it causes the ribosome to fall off. It just goes through, zooms so fast, it falls off. And it kind of disturbs the RNA polymerase, and so this causes the RNA polymerase to also detach. So this will stop transcription, and transcription will stop occurring. I don't know if that makes a lot of sense, so I'll just try and say it again. In low tryptophan levels, I like to imagine that the hairpin loop, it's quite a slow, cruisy process. The ribosome go through and translate it slowly. This doesn't disturb the RNA polymerase at all because it's going through and translating it very slowly, very carefully. In cases of high tryptophan levels, the ribosome will zoom through that loop and translate it very fast, and as a result, you're actually going to have the ribosome falling off. And I just kind of think of it's called like an earthquake, and it causes the RNA polymerase to also fall off. Therefore, transcription will be halted as well. It is quite a complex process, and what I really recommend doing is looking up the VCEE Bio FAQ document. So if you look up this document, it really discusses what you need to know about this tricky concept that is, oh, it's PA, sorry. Why did it not download? Okay, it did download. This tryptophan, or tripoperon. I think the tripoperon is probably the most complex process we'll discuss. That and CRISPR, I think. So it might be a bit harder, I think. So definitely go through and look at this VCEE FAQ document because it really explains what you need to know about the tripoperon. You can see that diagram is literally taken from this document here. It explains what you need to know about it. And it kind of goes through lots of really difficult areas of study or like topics that you need to know. And so making sure you read this and understanding what goes on is really important. So do recognize that tryptophan, or the tripoperon in prokaryotes, has transcription and translation occurring together at the same time, and it's really important that these processes kind of work together. And so if the ribosome falls off, this can actually alter the polymerase. The polymerase may also fall off too. Whereas if the ribosome goes through and translate it very slowly, then that polymerase can go through and transcribe it slowly and it won't fall off. Cool. So just kind of emphasizing here that attenuation is possible because transcription and translation occur at the same place in the cytosol. And this isn't possible in eukaryotes because they don't occur in the same place because you actually got the presence of a nucleus, meaning that transcription must occur in the nucleus and translation occurs in the cytosol or the RAF-ER. So as RNA polymerase moves to the operon, the ribosome can begin translating the mRNA even though the full strand isn't completed yet.